Transcription is the first of two steps in gene expression. The process involves copying a gene's DNA sequence to make an RNA molecule. The three substeps of transcription include initiation, elongation, and termination. In this video, we represented the nitrogenous bases with M and Ms. Cytosine is yellow, guanine is green, adenine is orange, thymine is blue, and uracil is brown.
The first step of transcription is initiation. This is when RNA polymerase binds the DNA with the aid of transcription factors. The top strand is the non-template strand, while the bottom strand is the template strand. It commences the process of transcription. It begins in the transcription unit in the promoter region, located about 25 units upstream at the TATA box, a sequence of TATA nitrogenous bases. RNA polymerase attaches and begins the process of opening up the double helix. In this region is found the information to determine the amino acid sequence. The synthesis of the RNA strand begins. Transcription factors, represented by Doritos, are a collection of 5 proteins. Their job is to mediate the binding of RNA polymerase. Thus, the transcription initiation complex is formed, made up of the 5 transcription factors and the RNA polymerase. Transcription can now begin, and will do so at a rate of 40 to 60 nucleotides per second.
Elongation. The second step is elongation, which is the process by which pre-mRNA is synthesized in a 5' to 3' direction. The RNA polymerase moves downstream along the DNA, unwinding the DNA and making the RNA transcript. Afterwards, the DNA strand reforms a double helix.
The third step is termination, a process that occurs in response to various end sequences and stop factors. The RNA polymerase unbinds from the DNA, and RNA polymerase creates the polyadenylation signal coded AAUAAA. This signals for the transcription process to finish. Downstream from the signal, proteins cut the RNA strand free from the polymerase, and it is released entirely from the DNA.
At the 5' end of the pre-mRNA coding segment, there attaches a methyl cap, which consists of a modified guanine nucleotide. This cap facilitates the export of mRNA into the cytoplasm by leading it through the nuclear pore. It also acts as protection and a buffer for the important coding segment. At the 3' end of the pre-mRNA coding segment, a poly-A tail attaches to the polyadenylation signal. This tail consists of 50 to 250 adenine bases, and again acts as a buffer for the important coding segment. The methyl cap, the polyadenylation signal, and the poly-A tail together make up the UTR, or untranscribed regions of the mRNA strand. These parts are excess and were not transcribed from DNA. Unlike the UTR, the section between the methyl cap and the polyadenylation signal are the coding segment, or the part that was transcribed from DNA.
Next, RNA splicing occurs. Splicing is the process by which introns, non-coding segments of the molecule considered superfluous genetic information and which interrupt the sequence of genes, are removed from pre-mRNA. This then allows exons, segments which contain coding information which will be reflected in the proteins, to become connected to one another. At the 5' end of the intron, there is a GU, guanine uracil segment, at the 5' splicing site, while at the 3' end of the intron, there is an AG, adenine guanine segment at the 3' splicing site. There is also an adenine in the center of the branch site, or the middle of the intron. SNRPs, or small nuclear ribonucleoproteins, play a key role in the removal of introns. Various SNRPs attach to the splicing sites and to the branch site, where they aid in first manipulating the intron upward, then cutting the 5' splice site, which then attaches to the adenine in the center of the branch site to form a lasso-looking structure called a lariat. Then the 3' splice site is cut, and the lariat intron, which will later degrade, is released along with the remaining SNRPs.
The exons then form a covalent bond, and the process of RNA splicing is complete.